All right, guys, now we're switching to eukaryotic gene regulation that's used in organisms like us. So we are leaving prokaryotes behind. Again, operons are only used by prokaryotes for the most part. Eukaryotic gene regulation looks very different. The first level of gene regulation in eukaryotes starts with the chromatin. Chromatin is the combination of DNA and the proteins that it's wrapped around. Those proteins are called histones, and the histones can get different chemical modifications that allow that stretch of DNA that's wrapped around them to be either expressed and on or silenced or off. Heterochromatin is chromatin that's transcriptionally inactive or off, and there are two different kinds of heterochromatin. Some heterochromatin is constitutively off, which means it's always off because it doesn't contain any genes. This includes places like telomeres and centromeres. Facultative heterochromatin, on the other hand, is heterochromatic in some cell types and on or euchromatic in other cell types. And it can switch between being either heterochromatin or euchromatin. Euchromatin is stretches of the DNA that are transcriptionally active or on or can be expressed. Histone modifications determine whether a stretch of DNA is euchromatic or heterochromatic. Histone acetylation makes the histones less basic or more negative so that they repel the negatively charged DNA, loosening the interaction between the histones and DNA and making it easier for RNA polymerase to come in and transcribe the genes in that region. In heterochromatin, the histones are often methylated. Now, a methyl group doesn't have a charge. However, it creates a binding site for other proteins that can come in and further compact and crunch down and block transcription from happening in that area. So histone methylation shows up in heterochromatin in regions that are transcriptionally inactive or off. In addition to chemical modifications to the histones, the DNA itself can also be chemically modified. DNA methylation happens on CG base pairs, C's next to G's, and this methylation mark often shows up in heterochromatin. So DNA methylation is also a silencing off mark. Because DNA methylation happens in these CG-rich stretches of DNA, it allows a mechanism for copying. So if we separate those black strands of DNA and synthesize some new blue DNA, the new DNA doesn't have the methyl marks, but there are enzymes that can read methylation on one strand of the DNA and add them to the newly synthesized strand. This allows for a mechanism for copying the DNA methylation so that it can be inherited between one cell division to the next or even across generations. Having these methyl marks locks those genes down as being heterochromatic or silent or off. So the genes that were off in one cell type remain off once that cell divides and produces new cells. Both DNA methylation and histone modifications are examples of epigenetics, heritable changes in gene expression that are not due to changes in the DNA nucleotide sequence, not different alleles, not mutations, but those chemical modifications that change gene expression. Once the DNA is opened up or euchromatic, now transcription can occur. In eukaryotes, the kind of default state for genes is not off so much as not on, meaning they're not locked off, they're not silenced, they're not repressed, they're not blocked, but they're not really turned on enough to do any good. This is different than prokaryotes. In eukaryotes, most gene regulation is regulated positively. It uses activator proteins to enhance or ramp up or increase transcription to robust levels to turn the genes on. There are different types of proteins that positively regulate gene expression in eukaryotes. We've already talked about transcription factors, which bind to the promoter that allow RNA polymerase to attach and do transcription. These are the general transcription factors that are necessary for all genes to be turned on. There are also specific transcription factors that turn on a subset of genes in one cell type and not in another. There are also activator proteins that bind far away from the genes that they regulate. They bind to stretches of DNA called control elements. Control elements are stretches of DNA that are binding sites for different proteins. So the promoter is one kind of control element. It's a proximal control element that's close to the start of the gene. There are also control elements like enhancers that are distal or very far away from the genes that they regulate. And these are both binding sites for activators or transcription factors that positively regulate gene expression. There is negative gene regulation in eukaryotes, but these silencers usually function by blocking the activity of activators and thereby blocking transcription. Once the RNA is transcribed, that RNA itself can be modified. Anything that happens after transcription is considered post-transcriptional control. And we already discussed adding a 5' cap, a 3' tail, and splicing by cutting out the introns and gluing together the exons. These steps help the mRNA leave the nucleus, stabilize the RNA so it lasts longer in the cytoplasm, and also allow the ribosome to attach so that translation can occur. The length of the 3' poly A tail determines how long that RNA lasts. The longer the tail, the longer the RNA lasts. The longer it lasts, the more times it can be translated and the more protein can be made. Translation itself is a pretty blunt tool for regulating gene expression. Normally, cells only regulate translation in starvation conditions when there's just not enough amino acids to make the protein. Although some translation can be regulated by binding of hairpins in the 3' or 5' UTR. More common is post-translational regulation, where after the protein has been translated, it can be regulated by chemical modifications, such as we've already talked about before, such as protein acetylation, methylation, phosphorylation, adding sugars or glycosylation, ubiquitination, etc. Post-translational control also includes cleaving proteins to activate them, and this is true for insulin as well as for blood clotting factors. And then how long the protein persists in the cytoplasm will also regulate it. If a protein lasts longer, it can perform its function again and again and again. If you want that function to stop, then you can degrade the protein. One way to degrade the protein is to mark it with a chain of the small peptide ubiquitin, which targets it for degradation in the proteasome. One more point of regulation that kind of comes in at the side is RNAi, or RNA interference. Small RNAs can complementary base pair with mRNAs and either stimulate degradation of the mRNA if it's a perfect match, or block translation from happening if it's an imperfect match. And some small RNAs can even go back and complementary base pair with the DNA, forming heterochromatin and blocking transcription itself. For more information on post-translational control of proteins and RNAi, check out the 2 videos that I also posted from outside sources. Last question I want to leave you guys with to think about is why? Why are eukaryotes so complex? Why do we have all of these levels of regulation? What's the benefit? Why can't we just have the on-off switch of the repressor binding the operator like bacteria? What is the benefit of having all these different points of regulation? That's something I want you guys to think about.